Alright, number 5. In the lock and key model of enzyme action, the enzyme active site is thought of as blank. The correct answer is D. A rigid, non-flexible shape that fits the substrate exactly, just as shown in the picture here. So this is the substrate, it's the key, and this is the enzyme, which is acting as the lock. So A key-like shape that fits into a pocket of the substrate surface is incorrect, because the substrate is the key that fits into the enzyme active site. B, an area of the enzyme that can adjust to fit the substrate shape. That's incorrect because the enzyme does not adjust and has to have an exact shape in the lock and key model. So B is describing the induced fit model. C, a lock that bars a non-competitive inhibitor from reacting. This is not correct because inhibition prevents substrates from fitting into the active sites. This is a complete opposite of an enzyme-substrate reaction. In an enzyme-substrate reaction, we're looking for catalytic activity. So this one's kind of describing enzyme inhibition. And E, a hydrophilic area on the enzyme surface. So this is not correct because it's not just hydrophobic. It can be hydrophobic or hydrophilic, depending on the substrate.
Hey Jana, I've been struggling on my biochemistry homework all day long. Can you help me with question number 6? It reads like this. The function of the enzyme-substrate complex is to provide an alternative reaction pathway that blank. Letter E reads, lowers the energy of the products.
No, that's not true, because in the enzyme-substrate complex, the energy of the products cannot be altered.
Okay, well then it must be this one. D says that it changes the possible product formed.
No, in order to change the product, you have to change the reactants.
Okay, so then does it change the concentration of the substrate?
No, that's also not true. The enzyme-substrate complex does not change the concentration of the substrate because the substrate is a predetermined factor before the reaction occurs. An alternate reaction pathway cannot and will not change the amount of substrate that is put into the reaction.
I think I know it. That means that it lowers the energy of the substrate.
No, because in the enzyme-substrate complex, the energy of the substrate cannot be altered.
Oh, I got it. That means it lowers the activation energy for the reaction?
That is correct. An enzyme lowers the activation energy of the reaction because an enzyme is a catalyst, and a catalyst lowers the activation energy of a reaction. When you add an enzyme-substrate complex to the reaction, it will lower the activation energy for the reaction to occur.
Can you explain that picture to me?
I just did!
Oh, okay. Well then, thank you very much. Brought to you by the socks.